Hello everybody, welcome back to the Biochemistry for Health Sciences channel. This is a channel where we connect chemistry as well as biochemistry with medicine. So today we are going to talk about plasmalogens. In the previous videos we have started talking about amphiphilic lipids, and we'll continue this discussion because plasmalogens are also amphiphilic lipids. Recall from the previous videos what we mean by amphiphilic lipids. Amphiphilic lipids are lipids that have a polar head, and one or more non-polar tails. So plasmalogens, you can see, have again the polar head. So this would be the polar head. And then we have the tails, non-polar tails. The two non-polar tails. So plasmalogens are also amphiphilic lipids. So if you look at the plasmalogen structure, this part here is glycerol. So it is a glycerol, there is phosphate. So it is a glycerophospholipid. And we talked about other types of glycerophospholipids in the past. But what makes plasmalogen very special, very different from the others that we talked about, is this linkage here that we call the vinyl ether linkage. So basically plasmalogen is a vinyl ether glycerophospholipid. So besides the glycerol, one of the carbons of glycerol here you can see is connected to phosphate, and connected to phosphate is either choline or the alcohol ethanolamine. And connected to the middle carbon here is a polyunsaturated fatty acid, one of the polyunsaturated fatty acids. And you can see this is connected by an ester. That's the normal way as which most fatty acids are connected to the glycerol, through an ester. And then on the other end of the glycerol chain is either a saturated or monosaturated fatty acid connected to this carbon through the vinyl ether linkage. And that's what makes this compound, or this class of compounds, this is not just one compound, could be many different compounds, because these fatty acids could be different. And this could be choline or ethanolamine. So this represents many different compounds. But what's unique about this class is this vinyl ether linkage. So what are the functions of plasmalogens? Let's look at the functions of plasmalogens. So first of all, plasmalogens are amphiphilic lipids with head and tails. So they have surfactant activity. So it's not surprising that they can reduce surface tension because they are surfactants. And because they are part of membranes, they are important in cellular processes such as vesicle fusion, where vesicles fuse with the plasma membrane. But this is what we see happening in exocytosis, endocytosis. This is how neurotransmitters are released from neurons and so on. Again, being membrane lipids, they can affect the biophysical properties of membranes. It seems like these plasmalogens tend to make membranes more rigid, probably because of this ether linkage. It allows tighter packing. So some parts of the membranes have different viscosity and different physical properties compared to other parts of the membrane. So these parts of membranes that are different from the rest, they're called microdomains. So plasmalogens seem to affect the microdomains, or they're involved in making microdomains. An example of microdomain is called a lipid raft, and lipid rafts are also very rich in cholesterol. So plasmalogens affect membranes, and since membranes are involved in a lot of processes such as cell signaling, neurotransmission, and so on. So clearly plasmalogens have a lot of functions that are similar to other amphiphilic lipids. Besides these functions here, due to the amphiphilic nature, there is another very important function of plasmalogens, and this is because this amphiphilic lipid has this ether linkage. Now, ethers are more sensitive to attack by free radicals. So basically, these plasmalogens, the ether linkage in these plasmalogens, in case there's some free radicals around the membrane, they will attack these plasmalogens rather than attacking other lipids. Okay, so free radicals, unfortunately, is part of our life. It's estimated that about 4% of all the oxygen you take in will become free radicals. So it's just part of our life, and our cell membrane and our proteins and DNA are continuously being attacked by free radicals. So these plasmalogens seem to protect by having this ether linkage. They protect other lipids from destruction. Not only do this ether prevent the destruction of other lipids, when these plasmalogens break down, their breakdown products also seem to protect the breakdown of other lipids. So this antioxidant activity of plasmalogens is kind of unique to these amphiphilic lipids. And again, free radicals is something we'll talk about a lot in future videos. Plasmalogens are found in many different cell types, especially rich in cells in the heart, as well as myelinated neurons. The synthesis of plasmalogens begins in organelles that we call peroxisomes. So the first 3 steps happen there, and then the synthesis is completed in the endoplasmic reticulum. The last 4 steps occur in the ER. Since peroxisomes are involved in plasmalogen synthesis, anything that is defective with peroxisomes, such as defective peroxisomal proteins, is going to affect the amount of plasmalogens in the cell, and that's going to affect the structure and function of that cell. And that impaired structure and function of the cell is going to lead to signs and symptoms of the disease. Example is the disease adrenoleukodystrophy, ALD, which is due to defective peroxisomal proteins that then leads to various neurological signs and symptoms. So this is very important in this series of videos for healthcare professionals. We need to connect molecules, which is chemistry, to the signs and symptoms of diseases, which is your medicine. Okay, so this is one of the most important skills healthcare professionals and students need to know for the present as well as for the future. And that's one of the objectives of these videos here, is to connect molecules to disease. Okay, and we'll do that a lot more in future videos. One of the examples of a plasmalogen that is worth mentioning for healthcare professionals is this one called platelet activating factor, which is shown here. Again, you see the head, you see the fatty acid connected to the glycerol by a vinyl ether, and then here you see not a fatty acid, this is a small sort of a fatty acid. This is the acetyl group. Okay, so very small, it could be called an acid, but basically it's a very small fatty acid, the acetyl group. So this platelet activation factor is found in many different cells, platelets, mast cells, all kinds of white blood cells, macrophages, even the endothelial cells lining the blood vessels. And it plays a very, very important role in the process of inflammation. Okay, it plays a very important role which can affect bronchial constrictions, change the dimensions and permeability of blood vessels, affect the sticking or adhesion of leukocytes to the endothelium so that they can go and attack the whatever microbes that's causing the inflammation, and they can attract and activate a lot of leukocytes. So very important mediator of inflammation. And again, inflammation is a topic that we'll talk about later. The biochemistry of inflammation, very, very complex topic, but extremely important in medicine. So we'll have hopefully a few videos on the chemicals or the biochemistry of inflammation in the future. So that is all for now. We will hopefully see you in the next video. I was told by Quilly that he should be back in these videos once we start with proteins. Until next time, have a good day.